female gametophyte. So how the female gametophyte will develop? If you see how the one megaspore can give the 8 nucleated and 7 cell stage of female gametophyte. Do you want to know the answer? Let us see our chapter, that is the female gametophyte or embryo sac. First of all, thank you for clicking this video. So let us start female gametophyte or embryo sac. Yes. So before we would like to discuss about what is the flower. What is a fascinating organ of angiosperms. So flower is a fascinating organ of angiosperms. So in this flower, how the flower is modified? So flower modified by shoot system. So flower can modified by shoot system by means of sexual reproduction. Now in this flower having 4 different orals. What are those orals? So flower can consist 4 different orals. One is calyx, corolla, androecium and gynoecium. These are the 4 orals. So let us see. Sepals and petals, stamen, this is the ovary, stills, carpels or pistil. Calyx, group of sepals we called as calyx, group of petals we called as corolla. Now stamens, androecium is the female reproductive structure in a flower, that is the androecium having this stamens, androecium containing the stamens. Now gynoecium is having the carpels or pistils. So in this carpels and pistils, flower is morphologically and embryologically marvels and site of sexual reproduction. So in this flower morphologically and embryologically marvels and sites of sexual reproduction. Gynoecium. Gynoecium. What we call as gynoecium? Gynoecium is the female reproductive structure in a flower, that is the gynoecium. So female reproductive structure present in a flower, that is the gynoecium. What is the pistil and carpels means? The female reproductive unit, it is called as pistil or carpels. So in this gynoecium having one or more pistil or carpels. So in this pistil or carpels, they are divided the 3 parts: stigma, style and ovary. So each pistil has 3 parts. One is stigma, style and ovary. So if you see the stigma and style and ovary, this is the stigma, style, this is the ovary. Let us see one by one structure. That is stigma. It is a part of pistil or carpel where the pollen grains are germinated. That is the stigma. You can search the standing platform of pollen grain. Here slender elongated structure we called as style. Pollen tube grow through the tissue of style. Pollen tube can grow through the tissue of style. Now ovary. Next third one, ovary. Ovary is a basal part and a bulged basal part we called as ovary having ovules. This ovary is having the ovules. Ovary is having the ovules. Now if you see ovary having one or several chambers, ovarian cavity we called as locules. What is the placenta? What is the placentation? Arrangement of ovules within the placenta, it is called as placentation. Now in this, what is the placenta? Where the ovules are attached within the ovaries arising from the placenta megasporangia, or you see this anatropous ovule. In this ovule structure is a small structure. It is attached by placenta by means of stalk-like structure we call as funicle. Next one, the hilum. Hilum is represented interconnection between the funicle and ovules. So this hilum represent the interconnection between the funicle and ovules. Now if you see this picture, so every ovule is having one or two integuments. It can protect. Every ovule is having one or two integuments. Envelope layers we call as integument. Fight. Integuments are encirculated of nucellus. So these integuments are encirculated of this nucellus except at the tip of micropylar end. This is the micropylar end. Micropylar end. So opposite micropylar end, basal part is there. This basal part we call as chalazel end. Chalazel end. So basal part we call as chalazel end, and these integuments are encirculated except at the tip of micropylar end. So if you see this nucellus enclosed within the integuments, mass of cells, it is called as nucellus. So what is the nucellus? This nucellus encirculated within the integuments a mass of cells, it is called as nucellus. So this nucellus having abundant reserve food material. This nucellus can provide nutrition for the development of embryo sac or female gametophyte. Generally single embryo sac or female gametophyte can develop from one megaspore through meiosis cell division. So how the female gametophyte or embryo sac will develop? When the single ovule is having female gametophyte or embryo sac. Ovule has a single embryo sac or female gametophyte is formed from megaspore through reduction division. How it is? What is the megasporogenesis? Formation of megaspores from diploid MMC, megaspore mother cell, to reduction division. This process we call as megasporogenesis. This is called as megasporogenesis. Now, so development of megaspores from megaspore mother cell through meiotic cell division, they are forming megaspores, daughter cells, for diploid daughter cell. Now, what is the monosporic development here? In this 4 daughter cells of megaspores, one is only functional. Most of the flowering plants, only one megaspore only functional. Remaining 3 are degenerate. If you see, these 3 are degenerate. One functional megaspore is developed to female gametophyte or embryo sac. So one functional megaspore is converted. One megaspore only developed to female gametophyte embryo sac we call as monosporic development. MMC through first meiotic cell division, they are converted into megaspore dyad. Megaspore dyad means 2 cells. So megaspore dyad. And second meiosis cell division, they are converted into megaspore tetrad condition. So megaspore tetrad. Tetrad means 4. 3 are degenerate. Remaining 1 megaspore is functional. So functional megaspore can develop to female gametophyte or embryo sac. Now let us see how the megaspore can develop to the female gametophyte or embryo sac. Spore, functional megaspore, through first mitotic cell division is converted into two nuclear embryo sac, and next second mitotic cell division this converted into four nuclear embryo sac, and third mitotic cell division and megaspore is converted into 8 nuclear stage. In this 8 nuclear stage of embryo sac, how the nucleus arranging in female gametophyte. We see the from this 8 nuclear stage of embryo sac, this can move to the egg apparatus at the end of micropylar end. In this 3 egg apparatus, two are synergids. One is egg cell. Total how many? 3 cells. So here this is the synergids. Remaining one is egg cell. They can move to the chelagel end to form a antipodal cells. 3 cells can move to the chelagel end. That is the antipodal cell to form a antipodals. Remaining two are, this is the polar nuclei. One is polar nuclei and remaining one is central cell. Remaining 2 cells can move to the nuclei, another one central cell. So in this synergids, cellular thickening, in these 2 synergids can form a cellular thickening micropylar tip. It is called as filiform apparatus. So this is the filiform apparatus. So cellular thickening, in the synergids, cellular thickening at the micropylar end will form. That is the filiform apparatus. What is the filiform apparatus function? So to guide the pollen tube enter into this synergid. 7. These 2 cells can move to the central, form one large cell. So in this large cell having the polar nuclei, 2n condition polar nuclei. So in this female gametophyte or embryo sac, how many cells are having here? This 3 and 3, 6 and 7. So 7 cells. And how many nucleus is there? 3, 4, 5, 6, 7, 8. So how many nuclei? 8 nuclei and 7 cell states. So in angiosperms, female gametophyte or embryo sac having 8 nucleated 7 cell states. Right? Nucleated 7 cell states. So why we call it as monosporic development? From this one functional megaspore is converted into the developed to the embryo sac or female gametophyte. How it is developing, we observed here clearly. So this is the monosporic development of female gametophyte.